Welcome again, students. So we'll discuss about the seed dormants and the seed germinations. So part 4. So today we're going to discuss about the metabolic changes formed during the seed germinations. So this metabolic changes involves the physiological and the biochemical changes during the seed germination. So first is the physiological changes. So in this the water uptake. So in the seed germination starts with the inhibitions of water by dry seed coat. Due to inhibitions of water, the seed coat becomes more permeable to oxygen and water and less resistance to outward growth of embryo. After inhibitions, the inner content of the seed increases in volume, thereby exerting pressure on the seeds. Seed coat, leading to rupture of seed coat. So the plumule and the radicle emerges thereafter. Okay. Next the respiration part. So after initiation of germination process, enormous energy is required. So more energy is required for various biochemical changes which are met through rapid increase in respiration rate. So the sucrose is probably the respiratory substrate in at this stage, which is provided by the endosperm. So in oil seeds and pulses, the lipid and protein respectively are converted into sucrose by suitable biochemical reactions. So next that is a mobilization of reserve material. So as germination process, there is a mobilization of reserve material to provide. So that is a building block for the development of embryo. Then we can say energy for the biosynthetic process, and the next is the nucleic acid for protein synthesis and the embryonic development. Now we compare with the biochemical changes. That is the first point, that is a nucleic acid one. So as the high concentration of RNA in the embryonic axis prescribes cell divisions. So due to more cell divisions, DNA content is increased. Okay. So next that is the carbohydrate. So during germination of this type, the embryonic axis secretes gibberellic acid into the allurin layers, which is found in the seeds, which causes synthesis of alpha amylase. So these carbohydrates, which is insoluble carbohydrates like starch, are the important reserve food of cereals in the endosperm. So during germination, starch is hydrolyzed first into maltose in the presence of alpha amylase and the beta amylase, and then maltose is converted into glucose by maltose 1. Okay, so this glucose is further converted into soluble sucrose and transported to for growing embryonic axis 1. Okay, so the next that is a lipid one. So in many plants like castor, bean, peanuts, we store a large amount of lipids or the fat, we can say, as a reserve food in the seeds one. So during germination, the fats are hydrolyzed into fatty acids and glycerol by lipase enzyme. So these fatty acids are further converted into acetals by the process of beta oxidation. So this acetal is further converted into sucrose by glycerate cycle and is transported to the growing embryonic axis 1. So this is again the process 1. Next is the protein. So some plants store protein as reserve food in their seeds. So proteins are hydrolyzed into amino acid by peptidase enzyme. So the amino acid may either provide energy by oxidation after, we can say, removal of amino acid, or may be utilized in the synthesis of new protein. Okay, and the last part, that is the inorganic nutrients. A number of inorganic nutrients such as phosphate, calcium, magnesium, potassium, etc. are also stored in the seeds in the form of phytin. Okay, so these stored nutrients are liberated during germination due to the activity of various phosphatase, including the phytase ones. So these are the metabolic changes found during the seed germination in the seeds. The next part, that is the role of phytohormone to improve the seed germinations. So the hormones play an important role in this during the seed germination also. So those plants are incapable or not capable to grow seed germinations form. But for that various hormones can be used. Okay, and with the combination of these hormones, the plants can be formed. A new germination can be formed. So the plants are characterized by producing various types of growth regulators that differed in their chemical structures and physiological action one. So they include auxin, cytokinins, gibberellins, abscisic acid, ethylene, salicylic acid, jasmonates, then brassionosteroid and sterolactonase. So each of these is found to play an essential role in the mediating plant defense responses against the stress one. So during the early phase of seed germination, in the germination, a decrease in jasmonate and salicylic acid contained and an increased level of auxin. Okay, so both jasmonate and salicylic acid act as a negative regulators of seed germination. So next, that is the auxins are considered to be regulators of seed germination process in cross-talk with the gibberellic acids, then abscisic acid, and that is the ethylene one. So in the brassionosteroid, the signal could stimulate germination by decreasing the sensitivity to the abscisic, that is the abscisic acid one. So this abscisic acid inhibits water uptake by preventing cell wall loosening the embryo and thereby reducing embryo growth potential. So by the combination of many one or two hormones or by some ratios, you can form a seed germination. A seed germination can be improved in many of these plants. Okay, so DNA of this integrity can be changed. Okay, as well as we can say protein and the DNA content can be changed by adding these hormones. So thank you very much.